Thank you to Twist for inviting me to talk about some of our very, very preliminary work using their new kit. And what we're trying to do is predict prognosis using cell-free DNA in uveal melanoma. My name's Aaron. I'm a postdoc at the Center for Precision Health at Cowan University. So luckily, Stephen and I schemed beforehand, and I'm not going to go over biomarkers or cell-free DNA because Stephen did such a wonderful job introducing that concept. So what I'm going to do is just introduce you to uveal melanoma very briefly so you understand the context behind what we're trying to achieve. So uveal melanoma is a very rare cancer of the eye, and this affects your choroid, your ciliary body, or your iris. In Australia, about 7.6 cases per million per year are diagnosed. So across the country, this is about 200 cases. And one of the very sort of interesting things about this disease is it has a very defined genetic pathway that is highly associated with prognosis. So during the pathogenesis of the disease, you gain an activating mutation to one of four genes in one of six hotspots. So that's GNA11, Q209, GNA-Q, Q209, and R183 for those two genes, PLCB4 D630, and cis-LTR to L129. Following this, there's a branching of the pathways, and and then the disease will go down one of three pathways, and that could be getting an EFX mutation, an SF3B1 mutation, or a BAP1 mutation. And having those mutations correlates with somatic copy number alterations, but also survival. So, for example, this is Kaplan-Meier from the TCGA paper of uveal melanoma quite a few years back. But essentially, if you have an EFX mutation, you have this good prognosis. If you have an SF3B1 mutation, you have an intermediate prognosis where you can have the disease spread up to 10, 20 years. And if you have a BAP1 mutation, essentially the disease will spread, and it's just a matter of time depending on how many copies of AQ you have gained in that tumor. And so liquid biopsy is possibly a very useful feature in uveal melanoma. So at the primary stage, while this genetic component can be very easily screened through the tumor, only about 30% of patients will actually have an intraocular biopsy. Now, that could be due to a number of factors, but one of them is because patients don't particularly want, as you can see on this image, patients don't particularly want this to happen to their eye. It can cause side effects such as vitreous hemorrhage or detachment of the retina. They're not super common, but it does occur, and this does worry patients. And Stephen very nicely described the uses of ctDNA. This is a minimally invasive approach. We can look at the genetic content of the tumor. And the downside, unfortunately, is sensitivity. So that's what you trade. You trade minimally invasiveness for sensitivity. And just to reiterate, these genes cover 99% of patients with 6 hotspots and 3 prognostic genes. So this is really perfect for ctDNA detection. We can design a very small panel that covers, so this panel is 30 kb. It's very cost effective, and it will cover virtually every patient without having to worry about very large sequencing panels that cover, 500 genes or something like that. This is a really great use case. So to look at some previous research that we have done. So this is looking at ctDNA at the baseline. So this is when people are diagnosed with uveal melanoma. What is the level of ctDNA that they have in their blood? And so when we originally published this, so we were the first to describe ctDNA in primary uveal melanoma, and we used a technique called droplet digital PCR. This is a very sensitive single locus approach. But the downside is usually for this kind of approach, you need to know the mutation. In our case, we did serial screening. So we tested each of those hotspots, those 6 hotspots. And what we found was that we could only detect ctDNA in about 23% of patients before they had treatment. And what we can see is that the level of ctDNA was not correlated with tumor size. And this is likely because uveal melanoma is a very slow-growing tumor. It doesn't have a lot of cellular turnover, so it's not releasing a lot of genomes into the blood. And it's also the eye. I haven't seen any literature on the eye being a particularly good spot for um, release of ctDNA. So we're working against a lot of factors to try and make sure the sensitivity is really high here. And then following our study, there's been a few others have published the detection. So it's all been very quite low. Now, one of the key factors for the treatment of uveal melanoma is the fact that all patients, or virtually all patients, I should say, will go and have radiation or radiotherapy. This could be, brachytherapy through a plaque. This could be stereotactic radiation. This could be proton beam irradiation. But most patients will have some form of radiation to that primary tumor. And there's been some really recent literature showing that radiation increases the amount of circulating tumor DNA detectable in the blood. And this is because the radiation induces cellular death. And when those cells die, they release their genomes into the blood. And then, in theory, you should be able to detect them at a higher frequency than you would without the radiation. So we can see that in from Francis et al., DeBrun et al., and Kim et al. And from Kim et al., we can see that the correlation of ctDNA is now correlating with increased tumor size. So not only does radiation increase the amount of ctDNA in the blood, but it also increases the amount of ctDNA in the ocular humors. So in this study by Im et al., they looked at the presence of ctDNA in the aqueous humor. So the aqueous humor is basically a liquid in the front of the eye. And patients that had ciliary body uveal melanoma after plaque could also have detectable ctDNA post-radiation, but not pre-radiation. So this is again showing evidence that radiation is allowing ctDNA to be more broadly used in these patients. So in our study, and I must note that this is a very early look at a first batch of samples, so it's not complete. The analysis is still ongoing and the data collection is still ongoing. But we had 31 uveal melanoma patients pre and post brachytherapy. So this is where the radiation therapy. We had during their and when they were in theatre, we had a single blood draw of 10 mil. And from that, we had a median plasma volume of 4.4 mils going into this. We used the QIAGEN circulating nucleic acid kit. This is basically the standard in the field. It's the kit that gets you the best yield of ctDNA. And we have some specific additions to the kit, such as the using carrier RNA, double eluting, and concentrating the DNA, because we want as much DNA into our reaction as possible. We want to see as many genomes as we possibly can. When we did the twist cell-free DNA library preparation, we had a median input of 24.5 nanograms up to a maximum of 30 nanograms of DNA. So we're really, pushing the amount of genomes we're screening in our kit. And then when we did the hybrid capture, we had 1600 nanograms per sample. So we're really getting a lot of genomes. Again, I'm stressing that because it's really important. You want to see as many genomes to increase your sensitivity. And then for sequencing, we started off at a target depth of 100,000 times. So this was the 62 samples on one lane of a 10B flow cell. Our bioinformatic pipeline is pretty standard. We used FGBio, BWA-MEM2, and then duplex consensus calling. So this is a 2-1-1 duplex. So this uses the alpha and the beta strands, which are basically the two sides of a DNA molecule, to collapse into one read. So it's very it's less efficient than molecular consensus, but you're increasing your specificity by doing that. And then our mutation pipeline is just mutate and pile up and fragmentomics. So the cohort characteristics for this sample, so I'm just showing these to show that it's a very standard uveal melanoma cohort, the ages, the sex ratio, the ages, and the sizes are all pretty standard. I would say the size on maybe the smaller side for this first batch of samples. And we know the risk of only about just under half of patients. So this is all pretty standard for uveal melanoma. So the first thing I wanted to show was the pre-analytical features. So in our study for the pre and post time point, we had a similar amount of input. So we were kind of limited by being able to have one tube of blood during surgery. So, sometimes you get varying levels of plasma, whether it's a higher hematocrit or the blood tube is slightly underfilled. You do get a different amount of plasma, and then that will affect the input. But between the two time points, there was no significant difference. Um, the percent cell-free DNA was also not significantly different between the two time points. And what the percent cell-free DNA is, and this is quite an important feature when you're loading your libraries, you want to have as many mono, di, and trinucleosomes in your reaction as possible. So when we load our kit, we base this on the 50 to 700 base pair quantitation of tape station. So and that ratio is showing how much of your total DNA content is the ctDNA, not lysed leukocytes or contaminating gDNA. So they were similar between the two time points, but very interestingly, post plaque, there was a significant, and these are all paired samples, of course, there was a significant increase in the amount of total cell-free DNA that we were getting from, the same sort of level of plasma. So we're now increasing the amount of genomes that we're able to extract from these patients at this just by choosing a different time point. And what that allowed us to do is to input into our library preparation a much higher level of ctDNA. So you can see here a median of around 22 for the baselines, which is still very high. That's still a very high amount of ctDNA, but we were able to increase that to 30 sort of median for the post-plaque. So we're able to screen more genomes at this time point, which is very useful. Just to look at some sequencing metrics, and I just selected a few just to show you the quality that we obtained from our sequencing. So our fold 80 base penalty was all well under 1.5, so that's really phenomenal, really great to see. And our selected bases were all above 0.9, so that's really, really great quality data. And when we looked at the mean coverage we have across each bait region, so basically this graph is the mean coverage by bait region of our panel. This is our 30 kb panel. I've then colored this by the amount of DNA input. And what we see is that for the duplex analysis, so this is a 2-on-1, this is collapsing a lot of molecules together. For 30 nanograms, we're getting a mean coverage of 3,646 at about 130,000 times raw depth. So we're getting really phenomenal molecule counts at the specified DNA input. And this is basically above the Twist white paper that they released. It's a really great result. So moving into the results. So for our patients, we detected ctDNA in 24% of cases at baseline, which then increased to 45% at the radiation time point. And if we take both time points into account, we saw ctDNA in 58% of patients, which is an absolutely brilliant result. If we look at the maximal allelic frequency, and what I mean by this is I've taken, for example, if they have a mutation in BAP1 and a mutation in GNA11, I've just taken the highest frequency. I will note they're about the same, but I've just taken the highest one for this graph. We see that there's a significant improvement in the distribution of ctDNA detection. So you see a lot of from the baseline negative values that then become positive at the post-plaque sample. And what you'll also see is that the overall level of ctDNA detected at this time point is higher. And I just want to highlight on the y-axis these actual range of values. So these are quite small tumors, and we're seeing mutation frequencies from maximum of 1.1%, but most of these are at 0.1 to 0.05%. So we're really detecting very, very low frequency mutations, which is really great to see. Now, for the prognostic mutations, we've detected them in 3 in BAP1, which is the poor prognostic class, 4 in SF3B1, and 2 in EFX. Unfortunately, we still need long-term follow-up to validate these, and I've also requested where available tumors from pathology. So I don't have any correlation between those features yet, but it will come in time. We also see that post-radiation therapy, we see that at baseline, the amount of ctDNA released is not associated with size, as I showed previously in a slide using droplet digital PCR. So it's good to have that sort of orthogonal validation of that finding. But then we find that once patients have had radiotherapy, ctDNA is now has a moderate significant association with tumor size. So the bigger the tumor is, the more genomes that are being released into the blood, essentially. So when they're breaking down and dying, there's more that we can see. So again, it's showing that by selecting a specific time point, we're able to improve our ctDNA detection. Now, moving on to maybe some more fragmentomic analysis. So the first thing I wanted to show was that nucleotide diversity. So nucleotide diversity at the ends of fragments has been shown to be different in cancer versus healthies. So I wanted to just explore whether or not we could start to see any differences in this. And I will note again, remember these frequency abundances are extremely low. This is 1% and under. So this is more just an exploratory analysis. So what I found was that at the pre and post time points, there was no significant difference in the Shannon diversity of these nucleotide motifs. But between negative and positive, there was a slight increase in the Shannon diversity of the nucleotide motif. And these are those motifs there. And so what this indicates to me is there's something going on, and I will explore this more in the future, but it's just as a kind of showing that the increase in genomes is probably associated with the increase in ctDNA that we're seeing from the radiation samples. But interestingly, we didn't see any difference between the proportion of short fragments between 150 base pairs and 20 base pairs, and proportion of long fragments between 160 and 180 base pairs at this pre and post plaque time point. So short DNA fragments are generally associated with ctDNA, and we didn't see an increase in that in radiation. But again, remember, these are very small allele frequencies that I have shown, so it might not be a detectable change, but it's still an interesting finding nonetheless. So, in conclusion, the Twist cell-free DNA library preparation kit and hyperfraction performed really well. I would say it performed at the white paper that Twist released or greater. We saw mutation frequencies less than 0.1%, and you'll note that the input we put into these reactions is very high. So we had up to 30 nanograms with a median of 24.4 nanograms input. So that's a lot of genomes that we're able to screen, and the improved library conversion of the kit has really helped get a lot of those molecules through to the final analysis. And a lot of some of these mutations were also seen pairwise between the two samples, baseline and following treatment, which is really good internal validation that they're a real mutation. The sequencing panel at the baseline matched our previous droplet digital PCR detection rate, and then utilizing the panel at both time points improved our ctDNA detection above baseline testing only. Of course, what that means, or detecting ctDNA at both time points, what that means long term, we're not sure yet. We still need a lot of follow-up, but that will come in time. We showed that radiation in treatment increased ctDNA levels, so this is always something that you should consider when using ctDNA is what time point should I take a sample to get the most information possible. And it seems like if you can get a sample post-radiation, that's really useful. We could detect prognostic mutations, but in general, it's less sensitive than the driver mutations. So the driver mutations are hotspots. It's much easier to validate those than, say, like an indel from BAP1, but we could detect those still. And ctDNA detection in primary uveal melanoma remains challenging because, as you saw, the frequency abundances are very low, and this is because of the anatomical location of the tumors and the actual size of the tumors. They're very small. And so going forward, likely testing larger tumors will yield greater results. So I just wanted to highlight, just before I finish, one of the other uses for this, not just in this primary stage, is really for monitoring for recurrence. So a few years back, we saw that ctDNA rose in patients prior to PET scans in a proportion of cases. So we can see that by monitoring serially patients, we're able to detect ctDNA and then detect relapse early. And so when we ran a study recently, we used droplet digital PCR on known mutations, and of course, only proportion of patients we know that because they only have a biopsy. So because of the really great sensitivity of this sequencing kit and panel, we think that using that in this context would also be incredibly powerful for monitoring patients for metastases before, like in concordance with radiotherapy scans, and I think that would really improve in that instance. So I hope what I showed made sense and what got you excited for ctDNA and uveal melanoma. I wanted to thank the patients and volunteers whose samples, without we wouldn't be able to do any of these studies, so we're really appreciative of all their donations. I'd like to thank specifically Dr. Tim Isaacs, who we've been collaborating for almost 10 years now. And again, without his help, we wouldn't have these samples to perform the study. Lab mates and Raw Perth theatre staff who are always amazing and very helpful. So thank you for listening to me.